All right, so the last screencast I ended with just looking at the structure of the neuron and the process which neural transmission is completed. And today I want to talk specifically about action potential and neurotransmitters. So when we talk about the parts of the neuron, we talk about the axon and how there's an electrical charge that travels down the axon and how it's generated by the movement of positively charged atoms in and out of a channel's membrane, the axon's membrane. If you look at the surface of an axon, it's selectively permeable, and there is an exchange between the ions in and out of the surface of the membrane. And you can see the positively and negatively charged ions on the screen. So we're taking a look at a cross-section of the axon right here. And the direction of the neural impulse toward the axon's terminals is that action potential. What you need to know is that the very first step of action potential causes this brief change in the electrical charge. And if it's strong enough, this produces depolarization and an action potential. So if it's just resting and neural firing hasn't taken place, it's considered in a polarized state. Depolarization takes place when the gates in the neighboring area open, and there's like this pump in the cell membrane, the cell pump, and there's an exchange between sodium and potassium, and it transports the sodium ions back out of the cell, which is kind of listed in this second picture. And then the third picture over here, as the action potential continues down the axon, the first section is now completely recharged. So the signal of polarization and depolarization is part of the action potential process. The ions specifically are sodium and potassium, and that exchange allows for the depolarization to take place. The threshold is the level of stimulation that's required to trigger a neural impulse. We like to use the toilet bowl analogy. If you think about a handle on a toilet, you can push down the level a certain way or a certain amount until it gets to a certain angle. And once it gets to a certain angle, the flushing process begins. That's the threshold. That's the level of stimulation required to actually make the flushing process begin. In a neuron, the threshold is that level of stimulation required to trigger action potential or a neural impulse. Once the neuron is depolarized, the refractory period is this short period necessary for the neuron to recharge and allow itself to fire again. So if you think about the toilet bowl analogy, it works in that you have to wait for the bowl to refill with water before you can flush it again. That would be considered the refractory period in that analogy. When you look at the properties of action potential, it's referred to an all or none response. So when the depolarizing current exceeds the threshold, a neuron will fire. If the depolarizing current fails to exceed the threshold, a neuron will not fire. So it's either going to fire or it's not. And if that threshold has not been met, the neuron will not fire. The intensity of an action potential remains the same throughout the length of the axon. In our book, they use a good example with like firing a gun. And when you push the trigger or you pull the trigger, it doesn't matter if you slowly squeeze the trigger or if you quickly squeeze it, the rate of the bullet remains the same. So the intensity of the neuron firing remains the same no matter if the threshold is soft or if it's an intense threshold. All right, so today our second learning target is going to describe how nerve cells communicate. We know that between the sending neuron and the receiving neuron is a synaptic gap. This is the junction between the tip of the sending neuron and the dendrite or cell body of the receiving neuron. There's a tiny gap Sir Charles Sherrington referred to it as a protoplasmic kiss, this resting pause in between the neural transmission. And that's how the synapse was identified. So you can see action potential. Here's the terminal buds of the axon, and this is the receiving neuron. You can see that across the synaptic gap, this is where the neurotransmitter is secreted, and it binds to the receiving neuron or the receiving cell body of another neuron. Neurotransmitters are chemicals released from the sending neuron. They travel across the synapse and they bind to the receptor sites on the receiving neuron. And that influences the generation of action potential. So you can see the chemicals in here, the neurotransmitters, being secreted across the synaptic gap and binding to the receptor site of the receiving neuron. Reuptake is referred to as the absorption process. It puts the brakes on neurotransmitter action, and sometimes what you see is the neurotransmitter, after being released across the synaptic gap, you only have so many receptor sites that take in the neurotransmitter. So some of them are reabsorbed into the terminal bud of the sending neuron. Reuptake basically means absorption. So let's take a look at how neurotransmitters influence us. There are several neurotransmitters we're going to go over. The first of which is called serotonin. And serotonin is a neurotransmitter that is linked with mood regulation. A lot of you are familiar with serotonin, and it's linked to depression. I'm going to talk more about serotonin when we get to a later slide. Another neurotransmitter is dopamine. Dopamine in excess sometimes will cause hallucinations in schizophrenic patients. Dopamine in small amounts oftentimes is found in Parkinson's disease patients. And they have, with not enough dopamine in their system, they have trouble with movement and muscular coordination. This is an awesome chart that shows you the function of the neurotransmitters listed in our textbook, but it also lists examples of when the neurotransmitter malfunctions. ACH is the neurotransmitter that we know the most, that we have the most of in our body. It is at every gap of any muscle or joint, and it allows for us to move. It enables muscle action. We know that ACH or acetylcholine is also linked with learning and memory. Specifically with memory, we know that in Alzheimer's patients, the ACH producing neurons deteriorate, and with the lack of that, they have links to memory loss and Alzheimer's disease. So dopamine influences movement, learning, attention, and emotion. Like I mentioned on the earlier slide, excess dopamine receptor activity is linked to schizophrenia. If you have an absence of dopamine, the brain produces the tremors and decreased mobility of Parkinson's disease patients. Serotonin, like I mentioned, affects mood, but it also affects hunger, sleep, and arousal. An undersupply is linked with depression, and Prozac and some other antidepressant drugs, some other SSRIs or selective serotonin reuptake inhibitors, which we'll talk about later in another unit, will raise serotonin levels and elevate mood. Norepinephrine is a neurotransmitter that helps control alertness and arousal. Arousal in reference to being awake and ready to take on whatever challenge comes your way. An undersupply of norepinephrine can also depress mood, just like with serotonin. Norepinephrine is very similar to epinephrine. We often hear of people who have allergies carrying an EpiPen, and norepinephrine, epinephrine elevate mood, arousal, and alertness. I shouldn't say mood, actually. That's more serotonin, but it can help kind of give you that readiness feeling, like you're ready to go and take on whatever comes your way. GABA GABA-aminobutyric acid is a major inhibitory transmitter. We're going to talk about agonists and antagonists in a little bit. Undersupply can be linked to seizures, tremors, and insomnia. Glutamate, oftentimes we find glutamate in sodium, like MSG or monosodium glutamate. It's a major excitatory neuron. It's an agonist. It's involved with memory. Oversupply can sometimes produce migraines or seizures. That's why some people who have chronic migraines, doctors will recommend that they remove MSG from their diet to see if their migraines decrease. So this is basically a slide showing kind of a lock and key mechanism that neurotransmitters bind to the receptor sites of a receiving neuron. So you can see how that kind of takes place. This is a cross section right here of a receptor site, a receiving neuron, and how the neurotransmitter can bind like a lock and key to the receptor site and cause changes within the chemical makeup of your body. We're going to talk about two. Agonists are the excitatory types of neurons. It's almost identical to the neurotransmitters, that it can actually mimic the same effect. Morphine, for instance, mimics the action of endorphins by stimulating receptor sites in brain areas involved in mood and pain sensations. So agonists, remember, excite. Antagonists inhibit, and oftentimes you think of in literature antagonists being the negative character. So if you can remember that, it helps you remember that antagonists block and inhibit neurotransmission from taking place. So just like with agonists, these neurotransmitters are so similar in structure that they can mimic neurotransmitters and block action. Curare poisoning, like in botulism, it paralyzes its victims by blocking ACH receptor sites involved in muscle movement. All right, so that is the end of the discussion or the screencast on neurotransmitters and action potential.